Good morning, it's March 5th and this is your daily brief in science. Here's everything you need to know. A recent clinical trial has made significant strides in treating blinding corneal injuries through an innovative stem cell therapy known as cultivated autologous limbal epithelial cells, or CALEC. This study, published in Nature Communications, has reported remarkable success rates, with 93% of patients experiencing complete or partial restoration of their corneal surface after 12 months and 92% at 18 months. The therapy has demonstrated a high safety profile, with only one minor adverse event recorded, a bacterial infection linked to chronic contact lens use. All participants showed varying degrees of improvement in visual acuity, highlighting the therapy's effectiveness. CALEC specifically targets limbal stem cell deficiency, a condition that depletes essential stem cells in the eye, hindering natural healing and often resulting in chronic pain and vision impairment. The procedure involves harvesting stem cells from a healthy eye, expanding them into a graft, and transplanting them into the damaged eye, effectively repairing previously irreversible corneal damage. Dr. Ula Jurkunas, the lead researcher, emphasized that this therapy offers hope where conventional treatments have fallen short, significantly enhancing patients' quality of life. However, additional trials are necessary to facilitate FDA approval, with the goal of making this promising treatment accessible nationwide. Future research plans include larger, multi-center trials and the exploration of using donor eye cells to broaden CALEC's applicability for patients with bilateral corneal damage. Currently, the therapy is limited to treating patients blind in one eye, as it requires stem cells from their healthy eye, but advancements aim to utilize donor stem cells for wider applications. Approximately 70% of patients experienced improved vision after 18 months, although these changes were not measured as a success criterion. This trial is notable as the first human study of a stem cell therapy for eye treatment in the U.S., involving collaborative efforts from institutions like Dana-Farber Cancer Institute and Boston Children's Hospital. The therapy represents a significant advancement in stem cell treatment, utilizing adult-derived stem cells rather than embryonic ones to repair corneal injuries. Researchers at Rutgers, New Jersey Medical School have made a significant discovery regarding a potent form of brown fat in genetically modified mice, which could potentially enhance longevity and exercise capacity. These mice, engineered to lack the RGS14 protein, demonstrated approximately 30% improved exercise performance and lived about 20% longer than their normal counterparts, with female mice showing even greater longevity. The genetically modified mice also exhibited signs of healthier aging, such as reduced hair loss and graying. They were found to be protected against various health issues, including obesity, glucose intolerance, cardiovascular disorders, cancer, and Alzheimer's disease. Brown fat is known for its ability to burn calories and regulate body temperature, and it plays a crucial role in enhancing blood flow to muscles during exercise. To confirm the benefits of this potent brown fat, researchers transplanted it into normal mice. These recipients experienced significant health improvements within days, contrasting with regular brown fat transplants that typically take 8 weeks to show effects. The overarching goal of this research is to enhance human lifespans by focusing on healthful aging, specifically targeting diseases associated with aging such as obesity and diabetes. Stephen Vatner, the study's senior author, emphasized the need for targeted therapies to address specific aging-related health issues, rather than a broad anti-aging approach. The research team is working on developing a drug that specifically targets improved exercise capacity and metabolism, with the aim of having a testable drug within a year. While current methods like deliberate cold exposure can naturally increase brown fat levels, researchers believe a medication would be a more practical solution for most individuals. Vatner expressed optimism about the potential benefits of enhancing exercise performance as a key component of healthful aging. A new study from Northwestern University is reshaping our understanding of how the brain processes speech. Researchers have discovered that Heschl's gyrus plays a crucial role in interpreting the melody of speech, challenging long-held beliefs about speech processing in the brain. Published in the journal Nature Communications, This groundbreaking research illustrates how Heschl's gyrus transforms prosodic features, such as pitch variations, into meaningful linguistic information. The study tracked brain activity in 11 adolescent epilepsy patients with implanted electrodes, allowing researchers to measure how pitch accents are encoded as distinct linguistic signals, separate from actual words. During the study, participants listened to an audiobook of Alice in Wonderland while their brain activity was monitored. The results revealed that Heschl's gyrus processes pitch changes as significant linguistic units, indicating that prosody processing occurs earlier in the auditory system than previously thought. Traditionally, the superior temporal gyrus was believed to be primarily responsible for processing prosody. However, this study challenges that assumption, suggesting a more complex architecture for speech perception. Co-principal investigator Bharath Chandrasekaran noted that this is the first investigation into how subtle variations in pitch convey meaning in the brain. Using high-quality intracerebral recordings, researchers found that pitch accents are processed as abstract representations, underscoring the unique linguistic capabilities of humans. Unlike non-human primates, humans abstract pitch changes into meaningful language cues, highlighting a distinct aspect of human communication. These findings indicate that the brain encodes pitch accents early in the auditory processing stream, which may have significant implications for speech rehabilitation and AI voice recognition technology. Potential applications include improving interventions for speech and language disorders, such as autism and dysprosody in stroke patients, as well as enhancing AI systems to better understand and replicate the nuances of human speech. Engineers at the University of California, San Diego have made a significant breakthrough in tracking the electrical activity of living cells using atom-thick quantum materials, specifically monolayer molybdenum sulfide. A recent study published in Nature Photonics reveals that these semiconductors can sense biological electrical activity with impressive speed and resolution, marking a notable advancement in the field of bioelectronics. This innovative all-optical technique utilizes the interaction of electrons in these semiconductors with light, particularly through the conversion between excitons and trions, to detect electrical signals. By harnessing the quantum properties of these two-dimensional materials, researchers can directly measure voltage changes in cells, providing a more effective alternative to traditional methods that often suffer from interference and inaccuracies. The new method allows for real-time monitoring of heart muscle electrical activity, achieving unprecedented speeds in mapping cell electrical behavior. By measuring changes in photoluminescence, the research team successfully conducted real-time mapping of heart muscle cell electrical activity at remarkable speeds. The study indicates that the high density of trions, formed due to sulfur vacancies during production, enhances the material's responsiveness to electric fields generated by living cells. This research has received support from multiple grants, including those from the National Science Foundation, the National Institutes of Health, and the Office of Naval Research, highlighting its significance. The implications of these findings could be profound, especially for studying neurological and cardiac disorders, potentially leading to advancements in treatments for conditions such as Parkinson's disease. The technology's potential applications include mapping dysfunctions in neural and cardiac tissues, improving understanding of diseases, and refining therapies like deep brain stimulation and cardiac pacing. The ability of these ultra-thin semiconductors to detect biological electrical signals with high speed and resolution is crucial for advancing our understanding of cellular functions. Researchers at the Icahn School of Medicine at Mount Sinai have made a significant breakthrough in diabetes research by developing a method to protect insulin-producing beta cells from glucolipotoxicity. This harmful condition is linked to type 2 diabetes, which affects over 500 million people globally and is characterized by high blood sugar levels due to insulin resistance or beta cell failure. Glucolipotoxicity arises from prolonged exposure to elevated glucose and fatty acid levels, leading to beta cell dysfunction and eventual death. The study introduces novel molecular glues that enhance the interaction between the transcription factor CBP-alpha and 14-3-3 proteins. These molecular glues prevent CBP-alpha from entering the nucleus under glucolipotoxic conditions, which is crucial for preserving beta cell function. By inhibiting the production of Cre-BP-beta, known to be detrimental to beta cell survival, these glues significantly mitigate the harmful effects of glucolipotoxicity in tests with primary human beta cells. This research is a notable advancement in diabetes treatment strategies, as transcription factors like Cre-BP were previously considered undruggable targets. Lead author Leora S. Katz, Ph.D., points out the potential therapeutic implications of using small molecules to fine-tune Cre-BP activity, which could lead to better diabetes management. Donald K. Scott, Ph.D., noted that this innovative strategy could complement existing diabetes therapies and help prevent disease progression. Unlike current diabetes treatments that primarily manage blood sugar levels, this new approach aims to directly prevent beta cell loss, potentially reducing the need for insulin therapy. Ongoing research will focus on refining these molecular glues for clinical use and testing them in preclinical diabetes models. The study was conducted in collaboration with teams from Eindhoven University of Technology in the Netherlands and the University of Duisburg-Essen in Germany, supported by various grants from NIH, ERC, and other European funding agencies. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.